First, I want to thank the organizers for giving me the chance to talk to this conference. Today I'm gonna talk about this note that I submitted in the proceedings, which includes information from two places, mostly from a joint work with my PhD supervisor, Matilde Marcoli, Gabor Frames for Contact Geometry in Models of the Visual Cortex. And also, part of the presentation is can only be found right now in my PhD thesis, but it will be soon uploaded in a separate paper on arXiv. And the purpose of the talk is to present a model of the functional architecture of the visual cortex, in particular of orientation sensitivity in the visual cortex, in terms of both its contact geometry and its signal analysis properties. And why one would want to have such a combination model is that if we do have that, then we can use a countable family of receptive profiles to encode and decode the cortical signal in a similar manner as Fourier series encodes and decodes a signal. And something that it's not so much of the scope of this session. In general, if we get inspiration from this, we can introduce a mathematical framework for signal analysis on a particular class of contact manifolds. The challenge was that the primary visual cortex has a smooth manifold structure, and on top of that, a contact structure. And so far, there haven't been any good constructions for filters on manifolds or a signal analysis framework on manifolds, except from some very special cases, for example, conformally flat manifolds. And the structure will be the following. I will roughly describe the two models we were based on. Then in the second part, I will present the model. And at the end, if there's enough time, I will mention some extensions of this. The first model is the one of the connectivity of simple cells of the visual cortex. Back in the 80s and later on during the 90s, from Hoffman and Petitot, respectively, the hypercolumnar structure of the visual cortex in terms of orientation was modeled using the manifold of oriented contact elements. A contact element is a tangent hyperplane at each point of the retinal surface. If we assume that the retina is isomorphic to R2, then the manifold of oriented contact elements is the product R2 times the circle, where the circle gives all the different orientations. And the important properties from this model that I would like to focus on is, first of all, this brings the fiber bundle structure on the visual cortex over the retina, where the base is the retina and the fiber is isomorphic to the circle, giving all the different orientations. And it also endows the model of the visual cortex with a contact structure, which is globally given by this contact form here, alpha, where here it's expressed in local coordinates, but it has a global meaning. And finally, the fibers of this contact fiber bundle are always horizontal to the contact distribution, which is what we call a Lesandrian circle bundle. The Lesandrian part refers to the fact that they are horizontal with respect to the contact distribution. So if we assume that there is a Riemannian metric on the retina, the manifold of contact elements modeling the visual cortex can be replaced with an equivalent contact manifold, that of unit covectors, where the contact structure will be given by the same form. The second model it has to do with the function of the simple cells of the visual cortex as filters. First, during the 80s, Daugman and Marsella used some specific kind of functions called Gabor functions to model the action of a simple cell that is centered on a specific point on the retina, x naught, y naught, and has a preferred spatial frequency omega and preferred rotation theta. And they based their results on the fact that the neurons minimize some uncertainty principles, so they localize at the same time the position and the frequency of the signal detected on the retina. So they use this psi function, where the first part of it is a Gaussian shifted in space, so shifted in the retina by x naught, y naught, times a complex exponential, so a modulation factor. If we fix the spatial frequency, then we can express this Gabor function using two unitary operators on L2. The TAF operator, which is the translation on the retina, and the M operator, which is a multiplication with the complex exponential on the frequency domain. So the question that arises is if one can use countably many orientation and position depending neurons in the form of these Gabor functions to represent and reconstruct the cortical signal, which would be a function from V1 to R. And the answer would be way more straightforward if we assume that the retina is flat, that it's isomorphic to R2. The answer would be coming from the 40s, where Dennis Gabor proposed a way to use a family of Gabor functions, so functions similar to those that model the simple cells of the visual cortex, to reconstruct a signal in L2 space into a series where the parameterization will be quadruples on a rectangular lattice as we see in the picture. The Gabor theory now has evolved, and they have been using way more complicated sampling sets, but for simplicity here I will just mention this rectangular kind of lattice. The family of functions parameterized by this lattice has to satisfy some properties in order to be good enough to make the series to converge. And this property is called the frame property, and it gives the name to the family of functions as well, called the frames, which is a weak Parseval identity, like the one from Fourier analysis. Instead of an equality now, we have a double inequality, where the lower and the upper bound are sufficient for the family to be complete, but most of the times this family will be overcomplete, which is a good property for a biological system, because it means that if we distract parts of the series, then we still have the convergence that we want, as opposed to a Fourier series. And nowadays we know that these models are not strange, like they are not different, like they are connected to each other. So the simple cells that appear to have this double nature as Gabor filters and as points in a Alexandrian circle bundle, they behave like this for a reason. And Sartine-City and Petitot used the action of the rototranslation group to relate the set of Gabor filters with the hypercolumnar arrangement and the connectivity between these cells. And that was more or less our motivation to see if one can introduce a Gabor analysis framework on the visual cortex determined by its geometry. And we think it does. So again, the answer would be much more easier if we assume flatness for the retina. But instead, we want to assume that it's a Riemann surface with a Riemannian metric. And the problem is that if one wants to define the filters for signal analysis in a Riemann surface with a Riemannian metric, we will stumble into the problem that we don't necessarily have a group law that will give us convolution products. So because it's not a vector space like R2, we have some difficulties, and we haven't also assumed some group structure. So a curved manifold like the retina doesn't necessarily have this group law. And the key observations here was that in order to overcome this, one has to lift to the tangent bundle of the retina, so use a consistent system of linearizations of the signals and the filters. That would fix the problem with the convolution, because now we would be on a vector bundle and the fibers would be vectors, but it wouldn't solve the second problem, where the filters, they can be functions on the tangent bundle of the retina, but they should be parameterized by both position and orientation. So if they were functions on the tangent bundle of the retina, they would be only parameterized by position. We need 4 building blocks to complete the framework. 1, to define an appropriate L2 space for the signal, a window function, a generalized version of the lattice, of a regular lattice, and finally a frame condition. So I'll start from the bundle of signal lattices, because we want the domain of the signal to be a consistent family of replicas of the tangent spaces of the retina. But we want the base manifold to be the visual cortex. Then instead of the tangent bundle of the retina, we use the pullback bundle of the tangent bundle of the retina on the unit cotangent bundle, which models the visual cortex. That way, the functions eat vectors of the retina, but they are actually parameterized by the both position and orientation. And a signal will be a function on this on L2 of this pullback bundle, the epsilon bundle, with respect to the volume forms induced by the Riemannian metric, where first we integrate fiber-wise and then we integrate for the whole manifold. So one would also want some consistency, because now with the lifted signal on the pullback bundle and the one originally being on the retina. So if we assume compactness of the retina, more generally, if we assume geodesical completeness, then one can determine the f function. Then the f function determines a lifted signal on the pullback bundle, and this can also the f function can also be recovered from the restriction. So, although eventually the Gabor analysis will happen of the fibers of the epsilon bundle, we also need some consistency with what we actually have on the retina. And then we want a window function. We started by defining defining a function that looks a lot like what we eventually want in the Whitney sum of the tangent bundle of the retina and the cotangent bundle, which takes a vector and a covector, let's say pointwise, and it sends them to this exponential, where the first part is Gaussian, a generalized Gaussian, with alpha being pointwise symmetric and positive definite. And the second part is a complex exponential, where the brackets denote the duality pairing between vectors and convectors. Now, if one restricts this to the sub-bundle, where the second part of the Whitney sum, the cotangent bundle, is just restricted to the unit covectors, the function is actually parameterized by xy and the angular coordinate for the fibers of the unit cotangent bundle. And this restriction gives us a window function on the pullback bundle where we want it to be. The third building block of Gabor analysis will be some sort of sampling set. So instead of using the classical lattices, now we are on vector bundles. So we had to introduce a bundle of lattices. If we assume that x and y are the local coordinates of a Riemann surface, and given the almost complex structure on the retina, here it's integrable due to its dimension, but it generally doesn't have to be. We just need an almost complex structure. We can obtain a twisted Liouville form, so on the cotangent bundle of the retina. So we have the standard Liouville form, and using the almost complex structure, we twist it a little bit. And this twisted form has the property that when restricted to the unit cotangent bundle, is also contact in the same manner where the restriction of the Liouville one form is contact. And the good thing is that now we have our model of the visual cortex, and it is a Lezandrian circle bundle with respect to both these contact forms. So the fibers are tangent to the distribution given by the restriction of the Liouville form and the restriction of the twisted Liouville form. And on top of that, the rib fields of each contact form, they are horizontal with respect to the contact distribution of the other. That means that when we project on the tangent spaces of the retina, we don't lose any information. 2 minutes? Okay. Okay, I'll go faster. So then we can use the rib fields as to generate the part of the lattice bundle for the pullback bundle, and the contact one forms to generate the lattice bundle part for the dual of the pullback bundle. Finally, the family of Gabor filters will be generated by these translations and modulation, as in the classical theory. And we say that they satisfy the frame condition if there are real positive and positive valued functions C and C prime over the local charge of our manifold, such that they satisfy the fiber-wise frame condition. We stumble on two problems about the lattice, which leads in some rescaling. One comes from the fact that in practice one can only measure finite amount of neurons, and so we had to cut off by the dark gray area. And also because we need consistency between the base and the total space of epsilon, we had to restrict into the area where the exponential is a differomorphism. So we had to use the injectivity radius of the metric in order to use this, to have this B, which was used to rescale the vector part of the lattice. And we ended up with this theorem, which says that this family of functions with the rescaled lattice is actually a frame, but the one with the non-rescaled lattice is not a frame. What do we get out of it? We get a series reconstruction and a representation of the signal. Here, I'm sorry for the notation, it should have been I instead of F. So the bottom line is that we replaced R2 with a Riemann surface and with a Riemannian metric to be able to account for the fact that the retina is not necessarily flat. The visual cortex in this case would be the unit cotangent bundle of this surface. The retinal signal will be a function from the surface to R. The cortical signal will be this lifted signal to epsilon. The lattice will be a bundle of lattices, and in particular, it can be degenerate, but it won't be degenerate in an open dense subset of them. And And I'll just say one thing. One can generalize in higher dimensions.
Your time? Okay, thank you very much. Thank you. Thank you very much for your interesting talk. Are there any questions?
In fact, I was interested in, we have 1 minute, about the thing in higher dimension. If what you did is can be generalized to higher dimensions. For example, with, I don't know, you substitute, I imagine, the Legendrian circle with some kind of Legendrian sphere.
Yes, we substitute the fiber with the Legendrian sphere bundle, and well, the generalization is not motivated from the visual cortex. So the construction is pretty much similar. We replace the almost complex structure with an action of the SON group, and we generated this a similar bundle of lattices in the higher dimensional case.
Okay.
So the hypothesis that you make is that over every point of the retinal plane, you have the entire set of Gabor frame, right? The entire Gabor frame. But actually, the pinwheels are very sparse. We have very few pinwheels, like 100 pinwheels. With respect to the points in the retinal plane, that is, I don't know how many, billions of points. So what happens if you sample in a very scarce way, as the cortex does?
Then indeed, if the sparseness of the pinwheels messes a lot with the density of the lattice, then it might lead to the fact that the theorem doesn't work. However, there is a lot of room in the lattice to be good enough to generate a frame. So basically, one needs to multiply with a less than one parameter, and the frame is done. So the answer would be, I would have to know exactly the sparseness to say, is it a problem or is it not? But the good thing is that they're very stable overall.
Okay, since there are no other questions, I would like to thank the speaker again.